obviously the DNA sessions that I've been delivering for the last 7 years now have been so popular, and I can't be thankful enough to the people that have invited me into their schools and the feedback that I've received, that the students have really enjoyed it. They've enjoyed me coming in, delivering a double period session for their students to have a hands-on practical lesson on DNA, fingerprinting, using the techniques of gel electrophoresis and PCR. And I know some of you have obviously had that experience where I've been in your schools. Some of you obviously would like me to be in your school, so contact me if you want to arrange a date for next year. Not a problem, I'll get you booked in and get that sorted out. But I've got some of the equipment here. I've got the PCR machine behind me, and I've got the electrophoresis tank. I bring in all the equipment, so the only thing that I ask you to provide is some distilled water and the use of your computer. That's it. You don't have to prepare anything else. And I spoke about that in the first session that I did for CERC, was going over the practical side of that lesson. There's a presentation that I can send to you. There's some information that I can send to you as well around that particular session. But I just thought I would show some of the equipment. Everything goes in one box. You get the students into 6 groups. They make up the gels. They make up PCR samples. They run the PCR machine. They put the samples into their gel and then put it through the electrophoresis tank. And then we basically get results in terms of crime scenes, paternity tests, cancer gene detection. We do COVID testing as well, because obviously there is a big emphasis. Everybody knows about PCR tests now, but understanding how you actually do these techniques, you can actually learn in the classroom. So you're doing real life practical lessons relating to obviously what's happening in the world. So it leads me on to the ELISA. A lot of people, when I go in to deliver these sessions, they ask, what else can you do? And I quite often get asked to do this on the back with the advanced higher students. I mean, it can be used for, I'm quite happy to work with any students, any number of students. The only stipulation is the kit, because I try to keep costs to a minimum with the DNA one, I don't ask you to pay anything. It's a free of charge session, but I do ask the schools to buy the little kits. So the ELISA kit is about £32 and it does about 10 lab groups, and all the contents are in there. I find that this is the easiest kit to use. It is so self-explanatory. It's probably, some of you already know Douglas MacDonald, who was a biology teacher in schools in Edinburgh. He's now retired. He does the Mystrica colorimeter as well, Douglas. So he's got a phenomenal mind. He has so many ideas, but he came up with this idea a few years ago. I know he's delivered sessions for CERC as well, and I know that obviously he will help and support with those sessions. But unlike Douglas, I can be around a lot more schools. He doesn't obviously go out and visit schools like I do. So if anybody wants a session on this, again, just give me some warning. I'll get you booked in, see when a date's available. It covers basically one period. You could deliver this within about 40 minutes. And the students basically work in pairs. They're given 2 samples, so they have a sample each. You also are going to put in the mix an infected sample, which has got the little red dot on it. So that's your infected sample, that someone within the group is going to have that infected sample. So you get them to work in pairs, and you get them to, there's a little worksheet within the box as well, so they can fill that in as they go along. But depending on the number of students, you're going to do, if there's a small group, then you do 2 transfers. If there's larger groups, then you start to do more transfers because you want to see it spread further. So the whole point of ELISA, if people know the ELISA test, it's an enzyme-linked immunoassay. It's the same as a lateral flow test, so you could use it for applications like COVID, for detecting antibodies if you have had that virus. It can be used for so many other applications as well, HIV testing, sexually transmitted diseases, drug testing, and food testing as well. So there's so many applications. I would come in and deliver it. You buy the kit beforehand. All you need is the kit, paper towels, scissors, pen, job done. And I take the students through it step by step. It's very, very easy to follow. It can obviously relate to real life applications, which is really good for the students. I love the fact that it can sit at school level. My background is microbiology and immunology, so I did this within my course, but I never did it to 4th year at university. So I just think it's phenomenal that the students get this opportunity to do microbiology, everything that I did at university, PCR and electrophoresis that I did at 4th year as well, and now Elisa coming into it as well. So it really gives them a taste and an idea as to what it's like out there in terms of careers, in terms of industry. It gives them skills to go into university or college, and it just gives them something new. A lot of people say it's quite nice to have me coming in, a different face, for the students to actually hear my background, because I'll give them an introduction on me, a bit about my career path from university to working in a lab, to working obviously within science education, and just to give them a sort of overview of that career path that I chose to take. So, and then I'll go straight into the presentation in terms of the theory. The students will usually have covered that side of it, but I'll go into it in a bit more detail, and I'll have an interactive session and ask some questions around ELISA. Obviously, you can get different types of ELISA testing. You can get indirect, direct, you can get sandwich. More of the science behind it as well, into the application side of it, and then it's straight onto the actual experiment itself. So you get a little titer well with the kit as well. Everything's supplied. You're going to label it up. So the first 2 wells are your 2 positive samples. The next 2 wells are the 2 negative samples. And then you've got 2 samples from each pair. Okay, so they would be labelled with your, the initials of each pair. And then you're just going to follow, first of all, you do the exchange with members within the classroom. So you've got your sample, you take a couple of drops out and you exchange it with your neighbour's sample, and that's the first exchange, and you'll put their initials in. Then you'll go to another pair and you'll exchange with another person in that pair, and that'll be a second exchange. So you just keep adding until, obviously it depends, as I said, on the size of the class as to how many exchanges that you do. If it's quite a small group, then you want to do just a couple of exchanges. If it's a larger group, then you can do up to about 4 exchanges. So once you've got your samples exchanged, then that becomes your sample, and you just follow the instructions through on the sheet. Everything is colour coded. So you've got your samples, your substrates, and your antigens, antibodies are all colour coded. There's a tip here and it's all marked. You've got a blue, red, yellow, white, and I think that's all the ones that you have to put in, and then you just have to do a couple of drops into each well, and you do the washing steps. You're supplied with a buffer wash, comes with it. You're going to obviously, after each point that you add the drops, you're adding obviously to get the antigen antibody complex. You're doing the washing steps in between each one, and you're just tipping it out onto a bit of paper towel, and then you're going on to the next sample, and you're just working your way through the samples until you get an end result. How this actually works is it's foolproof. Because Douglas is obviously a biology teacher, How it would typically work is the antigen antibody would bind at the end, and what he's done is it binds at the start, so it doesn't matter about the washing steps. You will get that colour change regardless. Other kits that I've used have been quite intense, lots of washing steps. The students get fed up and bored, and then they just sort of lose interest. Whereas this, at least they will get a result with this one. And the result that you get is you get a blue colour change if it's positive. So this was our positive controls at the start here, these two samples here. Then you get two clear samples, which is your negative controls. Then you've got your infected person, and then you've got your uninfected person there at the end. And you're obviously going to work your way back to find out who started the actual infection. So how you would do that is you would get everybody to stand up in the classroom, and you can work it back to the two people that actually started that infection. It's very, very easy to follow. You get, as I say, 100% results every time, instead of doing all of this practical and you have to fudge it at the end, which we all know and have all done. But at least with this one, you can guarantee you will get 100% results. And it's very straightforward, as I say. So there's a nice presentation. If anybody would like to have a look at this prior to booking, I can send it over to you. If anybody wants any more information on it, just give me a shout. I am more than happy to help. As I say, that's what I'm here for. I hope that was all right for everyone.